Yes, sir.
Show of hands. Okay,
Yes,
thank you.
sir.
All right, so in last class, I introduced you the concept of shotgun sequencing, which basically goes from taking similar DNA molecules, breaking them randomly so that you get overlapping fragments, preparing what's called cloning these fragments into a vector. And because there are many random fragments such as these, whenever you do this process, it's called preparing a library of different DNA fragments. Then one can pick the vector from a cell colony in which this vector has been cloned and sequence the amplified version of a single DNA fragment using Sanger sequencing. And that is what you would be, that is what people were doing before the advent of um, next generation sequencing. And these methods that I'm showing you here on the slide, they were therefore called the first generation sequencing. So this is the first way in which how people were actually doing DNA sequencing. So I showed you how this process was done to sequence small viruses and the idea of shotgunning or breaking the big DNA into small pieces and then using a computer to piece together the original uh, sequence was founded. And then again, it was used to sequence some other very highly prevalent molecules of DNA, such as mitochondrial DNA. And it is this at this point of time that people conceived the human genome sequencing project, where it was people asked that if we can sequence organisms which with about 100,000 nucleotides, that is 10 to the power 5, the same principles should ideally be applicable to sequence the entire human genome, which is 4 orders of magnitude higher, having 3 into 10 to the power 9 bases. So this is the idea, and it was sort of formalized in the form of the Human Genome Project, which was proposed by the US National Research Council. But it was not just the US, there were a lot of people from around the world who were leading genomicists at that point of time, including a person from India. His name was Samir Brahmachari. He was the director of IGIB in New Delhi, and a lot of people all across the world and India, they were brought together, and they formed 8 main goals of the project. The first goal, obviously, was to develop was to actually sequence the human genome. The second goal, which you can imagine at that point of time, because the sequencing technologies were first generation, so they were slow, so they wanted to increase the bandwidth, that is how much information you can create or exchange, and increase the speed with which you can increase which you can sequence. So this would mean that you would develop new sequencing technologies, which popularly came to be known as second generation sequencing technologies. The third idea was to identify human genome sequence variation. So human genome sequence variation is a concept whereby you say that the reason why two humans they look different from each other. So for example, all of us in class today, so we have been graced by 13 of you today. Um, so you and me, there are 14 people, and each one of us are different from each other, primarily and in large part because we have sequence variations amongst each other. So there are approximately 1 million, about 6 million locations in our genome where each one of us would be very different from each other. Given that you have differences between humans, it was proposed that you can take these differences and you can perform what's called functional genomics. That is, you try to understand how genomic variation, how mutations in the genome lead to functional changes. And you can do comparative or evolutionary genomics, which essentially means that how, given that you have the sequence of an entire organism, let's say a human, and let's say another organism like a chimpanzee or a mouse, you can compare and you can now find out the place locations or genes where there are mutations and how these mutations are different between different organisms. Given that this is
Sir,
something, yes,
sir, you said that like 6 million, there are 6 million places. So, are you talking about like 6 million nucleotides, right?
I'm talking about approximately 6 to 10 million nucleotides. That was the perceived number at that point of time. I'm talking about an average number, Ratnesh. Okay, I'm not giving you an
Yes,
absolute
sir.
number.
Sir, again, one more question. Like, sir, the thing is that in one video on YouTube, that was a TED talk. There was a person who was describing that like just by getting the DNA sequences, we can recognize the facial like face of the person. So, sir,
Yes.
are we able to get that kind of thing, or that is like still fictional thing? I
No,
just
that is
wanted to
definitely.
because.
Would you let me answer?
Yes,
Yeah,
sir.
that is definitely possible. And I can talk to you about how it's done. That falls under the purview of functional genomics. So what they do is they essentially have lots of faces and they have their underlying genome sequence. And then they ask the computer to learn what mutations in the genome are correlated with what kind of facial features. So there is a whole company, array of companies. One of the companies that I know is called FaceMap, who are doing this. So it is possible. So facial features are probably not as important, but I mean it is important when because there are a certain kind of certain kind of mutations that cause, say, a cleft lip. So when the lips, the middle line in the lips is joined, and these are the kind of so people start wanted to do this kind of work in order to recognize uh, what kind of mutations might be behind such a phenomena.
Yes, sir. But the question is that like last year in May 2021, we got the first complete human genome, and that is also not 100%. That is also like 99.1%
Good
or
observation.
2%.
Good observation. I'm going
Yes,
to talk
sir.
about why that is the case. But you have to
So then
understand,
how
Ratnesh.
can we recognize?
Ratnesh, can I finish? So there are
Yes.
15 people now in the class, right? If I have to differentiate between the 15 of you, do I need to know everything about each one of you?
No, sir. No.
Now, do you understand the answer to the question? So, not
Yes, sir.
everyone
Understood.
is going to
Understood.
be directly responsible for a feature, right? So, by if I want to distinguish between five people, I can just use five different features: their age, their height, their name, obviously, right? So that's the same thing with the genome as well. I don't need to know the entire genome in order to distinguish or in order to use the genome to extrapolate what that organism is going to or what that individual is going to look like or what kind of features the individual is going to have. This is a completely astute observation, and it's really good that you are reading about this, that the human genome sequence is not complete. So we cannot make the statement that we understand everything that the human genome or all the kind of information that the human genome has. But the information that we have is enough to actually tell us a lot of things whenever we are specially comparing to humans. Can I go
Okay.
forward?
Sir. Yes, sir. Yes.
So obviously, now it's interesting that you bring this thing up that given the face of a person, you can actually go back and find out what their genome was. Similarly, you can also imagine by knowing the genome of the person, you can also predict what kind of diseases the person might already have or may develop in the future. So this comes to a lot of these ethical issues or a lot of social issues. You must be hearing a lot of news around the current coronavirus being an engineered, weaponized version of the virus, right? And there was a lot of issues around it because it was being said that this has been used to target certain populations. While it has not been proven that it was the case, but it is now biologically possible to do this. So therefore, ethical and legal issues were really a big concern, and they are still a big concern. And countries like India don't have a policy in place to protect the genome sequence of individuals, specifically because the genome sequence of individuals, your DNA does not change. So it's the best, it's the most comprehensive fingerprint that you can have from anybody. Obviously, this was expected to generate lots of data. So there was one of the main goals, 7 goals, was to actually come up with algorithms and come up with computational pipelines and lay the foundation of what's called now bioinformatics or computational biology. And for such projects is always important to train manpower and people to carry on the project. So this shows you a small snapshot of what they were thinking. They would want to sequence the human genome or genomes, find genes. Once they have found genes, they would do genetic mapping or detection of mutations in these genes that are between individuals. Now, imagine that you have some individuals who are having a disease and some individuals who don't have the disease. So what you can do is you can separate the two people in the two groups, two kinds of people in the two groups, and you can ask whether people having a particular disease, do they have certain mutations in the genes? And does that those mutations actually correlate with the disease? Now, given that, what you can do, you can essentially say, okay, if a person has a specific kind of a mutation, and in those people with those mutations, you find a disease, then this mutation can actually be used as a diagnostic. So the example that I gave you in the first lab about BRCA1, BRCA1 actually is used as what's called a prognostication marker. So prognostication marker means what is going to happen to the individual in the future. And similarly, there are a lot of other kinds of mutations whereby if you have the mutation, most likely it's going to give rise to a cancer, or it is because you have cancer. Given that you understand this, establish a relationship between mutation in a gene and what is going to be the physical outcome, that is the disease, you can now find ways to cure the disease by attacking or by fixing the function of the gene to back to the wild type version of the gene, or the version of the gene that is not having any mutation. Now this leads to drug candidates. So basically drug candidates would basically be small molecules that can reset the function of mutated genes, or this can also lead to concept of gene therapy, which is now really very powerful now, where that is the ability to fix the mutations in your DNA itself so that the gene product now gets corrected, and therefore the disease which is manifesting or which is happening because of the mutation gets fixed. All right. I hope everybody understands. If you see this outline, you can explain the the goals behind the Human Genome Project. Yes, shall I move on?
Sir, can you briefly describe gene therapy? I have not know.
So
I do
gene
not know.
therapy
Like,
is the ability to change the mutations in a gene which is causing the disease. How it is happen, it
sir,
is outside the purview of the course. If you are interested, I can talk to you about it outside the class. But the idea is
okay,
that
sir.
if you have a mutation which is causing a disease, which is responsible for a disease, gene therapy essentially replaces that gene. So the therapy is happening on the gene. You're fixing the gene itself. Typically, whenever you are giving a drug, the drug is fixing the symptoms or it's replacing the effects of a gene, right? But if you have established that a specific mutation in the gene is causing the disease, then the best way to fix it is to fix that mutation. That is what gene therapy does.
Yes,
Anybody
sir.
else
Yes.
has any other question? All right. So it was all good that you want to sequence the human genome, but there is obviously lots of challenges. And I'm going to talk about all these challenges. So the first challenge is obvious. Before the human sequencing, genome sequencing project began, the largest genome that was sequenced was 1 lakh nucleotides long or 100,000 nucleotides long, right? Which is 4 orders of magnitude smaller. So if this was the original genome sequence, human genome was much longer. So length was a challenge. Can somebody tell me some other challenges? Quickly, please. Somebody wants to try? Want to try, then I will go for it.
Speed of the process.
Say that again.
So I said the speed of the process.
Yeah, speed of the process is not. I'm talking about the genome itself. So the other challenge is that earlier you were sequencing organisms, bacteria or viruses, which typically had only a single chromosome. And in the human genome, we know that there are 23 chromosomes, and each chromosome comes in pairs, one from your mother and one from your father, right? That basically means that even the DNA from each of the two chromosomes, although they look very similar, but the individual contents of the DNA. So, for example, if you take this chromosome number 19, they look very similar at the gross structural level, but the DNA sequence that comes from your mother is going to be different from the DNA sequence that comes from your father. And this is basically, for example, these red stars here show the places where the father's DNA is probably different from the mother's DNA. All right, so this is a challenge because now when you are sequencing, when you are fragmenting the DNA, right, there would be some part which would be coming from the mother, some part which would be coming from the father, and you would have to know what mutations belong to the father and what mutations belong to the mother, right? So this process of being able to place the mutations to the right chromosome is called phasing. You phase the mutations. The other problem, which is something what Ratnesh alluded to earlier, is the fact that the human genome is riddled with lots of repeats. So for example, if this was a bacterial genome, a typical bacterial genome, 95% of the genome has genes. In the case of the viral genome, the number is even higher. There is even the concept of overlapping genes in the viral genome. I can talk about it outside the class. But when you talk about the human genome, in the human genome, these same genes are going to be interspersed by a lot of repeated regions. And there are two major kinds of these repeat regions. One kind are called mini satellites, which are basically short 10 to 60 nucleotides, which are repeated 5 to 50 times, or they are called microsatellites or short tandem repeats. These are much smaller. These are 1 to 6 nucleotides longs, and again, they are also repeated 5 to 50 times. Now, this is how they typically look like. Now you will be interested to find it interesting that around 1984 to 85, when these repeats were discovered by Alec Jeffreys, who's now called Sir Alec Jeffreys in the UK, uh, he actually used the these repeated microsatellite and mini satellite patterns to actually fingerprint a human being. So basically, what he found was that each human had a fingerprint or had a specific number of such repeats. And these repeats could be used to actually identify the human being. And
Sir.
he laid the foundation for DNA fingerprinting, or what's now called DNA forensics. If you are interested to read more about it, I have given a link here. It's very, very interesting. All this kind of crime stuff happens using this ability to look at these microsatellite patterns in different humans. So for example, I'm showing you here a pedigree. So this is a father, the circle is the mother. And now when you have three children, you see that 16, 17, and 18. And now you take the DNA and you run the DNA on a gel to specifically look at the microsatellite regions. Now you can see that, okay, when you have the DNA of the children it's going to be a combination of the mother and the father. So a lot of times this DNA forensics was initially used to determine paternity. So a lot of these paternity tests, they are still actually following things like DNA fingerprinting using a microsatellite or mini satellite repeat regions. Yes, Ratnesh.
Sir, the thing is that RFLP can be done in human or any other eukaryotic because there is many satellite, microsatellite, but can it be done in bacteria also? Like today in the class of environmental biotechnology, Saroj ma'am was talking about there was paper as she was describing that in that paper that people have done RFLP for bacteria. So I was like kind of very new thing for me. Yeah.
So RFLP is the concept of
And that
using the.
for 16S RNA, 16S RNA. Yeah.
So RFLP as a technique basically relies on the fact that there are going to be regions where a enzyme can cut, the restriction enzyme can cut, right? So it's called restriction enzyme, restriction length, restriction RFLP. So
Restriction
restriction
fragment
fragment
length
polymorphism.
polymorphism.
Yeah, restriction fragment
Yeah.
polymorphism,
Length
uh,
polymorphism.
length polymorphism, right? So it basically means that if you cut a DNA with a restriction enzyme based on the repeated appearance of the restriction enzyme site, you're going to have a specific pattern, just like I'm showing you here, right? So the ability to do RFLP purely depends on the ability, your ability to cut DNA. So if you will have a restriction site and you have an enzyme to cut out this restriction site, you can do RFLP for almost any DNA fragment in the world. It doesn't matter if it's from bacteria or it is from a eukaryote. Typically, the enzymes that you have do not cut bacterial DNA because bacterial DNA, these enzymes are from bacteria. So the bacteria doesn't want to cut its own DNA, right? Because restriction enzymes are used by bacteria to protect itself against foreign genomes, such as viruses from bacteriophages. So but it's possible that if you have the right combination of an enzyme and that enzyme does not, and you have a site for that enzyme on the DNA of the bacteria, you can do RFLP in bacteria as well. Okay, moving on. I
Yes,
hope
sir.
these challenges are clear in the Human Genome Project to everybody. Okay. All right. So now, given that you have these challenges, the researchers came up with two solutions to sequence the human genome. The first solution, which was taken up by NIH and most of the academics, was called hierarchical shotgun sequencing. Okay. So the idea was to divide and distribute sequencing to multiple sites. The other approach that was taken by a private company called Celera Genomics was called whole genome shotgun sequencing. So the idea was you take a human genome, you randomly fragment it, and you sequence it on in one site. So can somebody quickly tell me if what challenges could be there in hierarchical sequencing and what challenges could be there in whole genome shotgun sequencing? Quickly, please. If you want to answer, answer quickly because I have a lot to cover. Nobody wants to give it a try? So something about repeats. So the question is when you talk about uh, human genome having lots of repeats, now how do you actually, so if you do shotgun sequencing and you just randomly break the DNA, the regions in the genome where you have lots of repeats, you might not be able to overlap it together, right? I think somebody in the class asked that question also earlier. Ashi, probably it was you, right?
Yes,
So
sir.
basically, so that's one challenge. So that's probably why whole genome shotgun sequencing might not work off this thing, just like that. So you have to think about it, how to work it out. And the other question in terms of hierarchical shotgun sequencing is how do you actually divide the genome? Is there some way to divide the human genome in a way that you can keep track of it? Can somebody think about it? Given that I have talked to you something about identification of human genome
Sir,
previously.
we know about like primer walking. So like if I determine that one part, then I go to that next part of that using a primer.
Yeah, but you have to sequence the first part, right? To know to design the primer.
Yes,
But
sir.
at this point of time, you haven't
Yes.
sequenced that part. The whole idea is that you have lots of DNA fragments and you want to distribute these DNA fragments across different laboratories of the world. Given that I have talked to you about certain challenges, can one of those challenges be used to actually identify and distribute a DNA fragment? So I'm going to talk to you about how that works out. All right, so this is the idea of whole genome shotgun sequencing. You have the DNA molecules, so three DNA molecules, identical DNA molecules that are randomly fragmented. Now you randomly fragment the entire human genome and you make into small Sanger sequencing compatible fragments. You clone these fragments, you amplify these vectors and you pick each one of these clones and you Sanger sequence the fragment completely, and then you overlap them. Obviously, this is not going to be completely viable solution because there are a lot of regions in the genome that are repeat regions. So the hierarchical shotgun sequencing actually does distribute the fragments based on the physical identity of the fragments by doing a physical map, by creating a physical genetic map. So how do they do it? We know that there are a lot of microsatellites that are appearing in the human genome. So they actually use the microsatellite repeat patterns to is to identify a physical map of the human genome. So how did they do it? They fragment the human genome into large fragments now. Instead of small fragments, they fragmented in large fragments. Now these large fragments are also overlapping, but each of these fragments was now cloned, okay, in a vector. And because of these fragments being very large, they had to come up with biochemistry or engineer vectors that could harbor much larger fragments. So these vectors were called bacterial artificial chromosomes or yeast artificial chromosomes because they were actually so big they looked like chromosomes inside the cell. And once they had done this, what they were doing was they were taking these artificial chromosomes or these clones and they were actually creating a physical microsatellite map of these. Okay, so let us say originally this was the, I'm just giving an example, this was the original um, DNA fragment that you, the genome that you had. Okay, there are two versions of this genome, two copies exactly. Now what you do is you randomly fragment these, you clone them in a yeast artificial or a bacterial artificial chromosome, and then you actually also do a microsatellite repeat mapping. Microsatellite repeat mapping can also be replaced by RFLP as Ratnesh was talking about. So once you have this microsatellite mapping, you already see certain, I hope you can see certain patterns. You see that these three repeats are also coming up in another fragment. So most likely they are overlapping, right? Similarly, these two close by repeats are coming up in another fragment, right? So like this, you can actually overlap these and you can make what's called a contig of overlapping sequences or clones. Now, what they were doing was once they had such a physical map of the human genome cloned into large chromosomes, large artificial chromosomes, they essentially took these large artificial chromosomes and they distributed across labs of the world. So typically most of this work was done in the US, the Europe and Japan. So that's why I'm showing you it's two examples here. So for example, the locus 1, 2 and 3, which was a contiguous locus, was sequenced in US. Then some locus which was the further locus that was coming up. So locus 4, 5 and 6, for example, was gone to sequencing in the UK. Right
So I had a
now,
question.
yeah.
So like you mentioned like in whole genome shotgun sequencing, like they make fragments of like 200 to 500 base pairs and they clone those fragments into like sequencing vectors, right?
Yes.
So like you'll have like probably like millions of fragments.
Yeah.
So
Yeah.
do they make millions of clones?
Yes. They had a
So,
whole
sir,
institute to do this thing. But obviously, so obviously they were not able to do this entire thing on their own. And at the end, what happened was, so the hierarchical shotgun sequencing people, they did this. So they fragmented, they distributed the ordered fragments across the world. So each lab would receive one of these clones, they would amplify the cloned insert, and then they would again randomly fragment, clone them into smaller, like into smaller DNA fragments and clone them and amplify them and then Sanger sequence, right? And what actually happened was these people, because they were academics, they were releasing these sequences very often. Okay, so they would release the sequence of the clone, right? Understood. So now given that this sequence was released, they could use this hierarchical shotgun sequences, these large parts of the DNA, human DNA cloned, as a backbone to align, to basically take the small fragments that the for coming from the company and actually uh, complete the human genome. So therefore the private company was using the public data and they uh, actually sped up the entire process of sequencing. So the real sequencing of the human genome was a combined effort of both the academicians who were taking this hierarchical approach, because initially it was said that the human genome is going to take 30 years to complete, sorry, 20 years to complete. But because of this shotgun sequencing approach, which was slightly faster and random, and the data sharing that was happening, they were able to complete it in 10 years only in 2001.
Sir?
So this is how it specifically looks like. You have hierarchical shotgun sequencing. Yeah, Ratnesh, maybe I can complete and then you can ask the question, Ratnesh.
Yes, sir. Yes.
Yeah. So you had the hierarchical shotgun sequencing where you had the genomic DNA, you broke it down into large pieces, and you made what's called a back library here. So back library is a bacterial artificial chromosome library. You call it a library again because this is a library. There is basically lots of these overlapping fragments. You organized mapped large clone context. So you basically, based on some physical aspects, you map these contexts into some kind of a linear arrangement that would represent their original location on the genome. You then tick one of these fragments to one lab, which amplified it, which again made shotgun clones out of it. You get the shotgun sequence. Now you overlap the shotgun sequence and you assemble. So how does it look like? You essentially, what you do is you have lots of these clones, and then you take a few clones out of this shotgun clone library, which called fingerprint clones. So you don't have to sequence all of them. You just have to sequence a few, and you can then get the entire back assembled. Once you have the entire back assembled, then you can actually then overlap them and merge data, and you can come up with much larger chromosomes or much larger lengths of human DNA that was sequenced. Is this clear to everyone?
So also, how efficient is this process? Because if I consider like people are making millions of clones, then like I would assume that there's an equally high possibility of there being some sorts of mutations or like errors. So if there are like mutations in some
Yes.
sequences, then how do they even superimpose them at
Good question.
together?
So therefore, what people were doing there was a concept of coverage. So that essentially means when you say that there was a X-fold coverage, so the human genome that we have today is approximately 65-fold coverage. Not actually today is much more, is about 300-fold. But at that point of time when it was released, each base pair was covered by 65 different clones. All right. So just by mistake, if one clone had a mistake, then you would say that at that particular location, I'm going to take the base pair, right, which is having the highest frequency of occurrence across all the clones, all the 65 clones that have been sequenced. Is this clear?
Right, sir.
So for example, if I look at this position here, I'm showing you, right? Here you can see there's 1, 2, 3, 4, 5, 6, 7, 8, 9 different clones which are overlapping at this particular location. Yeah. So when I say 65-fold coverage, it means that each base pair was a part of 65 different clones across the genome that was sequenced. That's why this was a huge effort. Lots of laboratories in the world, their whole job was to sequence like these back libraries for like 5 or 10 years. Okay, Ratnesh, you have a question.
Fine, sir.
Yes, sir. Instead of using PCR, why did they use for like cloning? Like for you have to make lots of copies of this DNA fragments. So why did they use clones of bacteria? And why didn't they use the PCR
So
for distributing?
so for PCR, you need to know the sequence, right?
Yes, sir.
So if you don't know the sequence, then you cannot use PCR. What cloning allows you to do is, in cloning, you insert the fragment in a previously known DNA fragment, right? So you know the sequence of the vector, the plasmid, and inside that plasmid you are putting the clone, right? So now you
Yes,
can
sir.
use primer walking and all those things that you were talking about. You can use the known sequence in the plasmid to design your primers.
Yes, sir.
Understood?
But in case of PCR, also for doing PCRing, like
You
making
cannot do PCR
amplifying.
without knowing the sequence, Ratnesh. Can you?
Yes, sir. Like primer is a very like 15 to 7, 20 base pairs, and that can be first known. And I guess
How? So I'm giving you a DNA sequence which is 200 base pair long. Okay. And you don't know the sequence. Can you design a primer for that without knowing its sequence?
no, sir, no.
Now you understand? So by cloning
Yes,
it,
sir,
you
understood.
actually, yeah, by cloning it, you put it in a area whose sequence you know. So at least if you know, I don't think you understand the concept of primer walking properly. We can talk about it, but at least by putting it in a clone, by putting it in a backbone, in a vector, you can actually know at least one part in the process of cloning. All right. So this
Yes,
is
sir.
what the idea
Sir,
was.
one more question.
Yes, Ratnesh.
So, sir, like you said that like did at the end NIH that Human Genome Project also collaborated with company? Like I think
Yes.
I have read
You
like
don't think
these
you
two
don't
were
think, no? I mean, see, Ratnesh, when you say I
competitive.
think, you should not be wasting my time by saying I think. If you know, you tell me that I know. So I'm telling
Yes, sir.
you,
I know that.
it was a part of two processes. So this was the Human Genome Project and the company is called Celera. And together there were two papers published. One paper was published in Nature, the other paper was published in Science. One was headed by the NIH consortium and the other one was held by the company. If you have found something otherwise, please write to me and I'll be more than happy to discuss it. Okay.
Okay,
So
sir.
if you want to understand the entire process, here is a video again by TED Talks. So it's a very small video. It very nicely explains exactly how the entire process happened. And yes, confirming Ratnesh, your question, there were two parts. There was a private company and the public partnership of the NIH driven companies. So this was the findings of the Human Genome Project to briefly tell you. So it was started somewhere around after 1985, after PCR happened. So a lot of scientific debate started to happen, and the consortium was formed in 1988 with the goals, and then from 1990 they started to sequence. So during the process, because they were sequencing this naturally, people also started to sequence not only the human genome but also other organisms, as you can see in red. So they started to sequence a lot of bacteria. So in 1995, approximately, you had the Haemophilus influenza bacteria genome that was sequenced. E. coli was sequenced in 1997, 98. You had another model organism, yeast, which is called Saccharomyces cerevisiae, that was sequenced. Then there was another model organism slightly bigger than yeast. It's a worm with about 1,000 cells. It's called C. elegans. Then you had another sequencing project going on for the fruit fly, another model organism called Sophila melanogaster. Their sequence was also sequencing also began, and then a model plant organism called Arabidopsis thaliana, which is basically a very small plant that people use to sequence to do experiments on plants in laboratories. Simultaneously, you had the mouse sequencing project happening and the human genome sequencing project happening. And as you can see, the first part that was completed from these projects was the genetic maps or the physical maps of the project. So basically, first about till 1995, they were creating a map of microsatellite regions and physical maps of the genomes so that you can take different fragments and can distribute it across the world. And you can see that this distribution actually started to happen after the microsatellite mapping have finished. So microsatellite mapping finished approximately in 1996, and it's only after that you started to distribute these fragments, these back clones, and then you had different time points at which the project finished. So first they sequenced the smallest chromosome, which is chromosome 22, chromosome 21, and then after that the project took to speed and it was published. In between, they also had something called sequencing of cDNA. So cDNA essentially is called complementary DNA. So you take all the RNA that was expressed by the cells that they were taking. So in the Human Genome Project, they were taking blood cells of 15 to 16 different individuals. They were mixing these blood cells and they were isolating the white blood cells from these uh, patients or from these people, not patients. And from these white blood cells, they actually also extracted RNA. They extracted RNA, converted this RNA into DNA by a process called reverse transcription by using enzymes that are present in viruses called reverse transcriptases. And they did the same process. So they took these complementary DNA fragments and they also made smaller fragments out of them and sequenced them. And these are often called ESTs or express sequence tags. Okay. And this is how the data was often represented, where you had basically a chromosome like this, and each of these dots actually represents one of the back back chromosomes, so back plasmid sequences that were sequenced. All right. So these were the findings. Briefly, they found initially that there were 2.85 billion bases in one set of chromosomes. So one set of chromosomes is called haploid. It's half. Or the complete set of chromosomes, which basically you have two chromosomes, is called diploid. It's two sets. It was initially estimated there were 30,000 genes. Later, it was revised to approximately 20,000 genes. And now it is widely accepted that there are approximately 18,000 genes in the human genome. So this is
Sir,
yes, Ratnesh.
about number of genomes, like how, where do we find, like on that complete human genome sequence, I see that something about 54,000 it is. What it is written, I'm not then understanding perhaps.
What is written where, Pratnesh? Where did you find it?
On, yes sir, on NCBI complete human genome, and that data, when I click and try to view, and there it is written that like number of genes something.
So this is a figure that is always revising. So people are not really sure, but they have a ballpark figure that approximately 20 to 50,000 genes are there. And where I'm talking about genes, I'm mostly talking about protein coding genes. I think the 54,000 genes that they mention also contains a lot of non-coding genes, non-protein coding
Maybe.
genes. Not
Oh,
maybe, I'm telling you.
yes.
Yes, that's the case. So this is only 1/4 as great as previously thought. So previously people thought that there were many more genes because there were many more proteins that were getting made. And this is potentially because of alternative splicing, that same number of genes can actually make many more different kinds of proteins. Um, and one major thing that was found was that less than 2% of the genome codes for proteins. That is coming from these 20,000 genes. An average gene consists of about 40,000 bases, but the gene size is very, very large by a large amount. You have a gene called dystrophin, which is playing a role in Duchenne muscular dystrophy, which approximately has 2.4 million bases, just a single gene. They also came up, as I told you, the proteome or the collection of different proteins in the cell was much larger than the genome. And therefore, they were estimating that an average gene product makes 3 different proteins. And the function of many genes still remains undiscovered. Because they were sequencing 13 different individuals, they saw that 99.9% of the individual DNA was shared between the individuals. Chromosome 1 had most number of genes, chromosome Y had fewest number of genes. Also because their sizes are much different. Chromosome 1 is the largest, chromosome Y is the smallest. So human genome was estimated to have approximately
Sir,
10 billion single nucleotide polymorphisms or mutations between two individuals. Yes, Ratnesh.
so chromosome Y is not the smallest. Instead, chromosome 22 is the smallest chromosome. Y is bigger than like many of the chromosomes.
Okay, yeah, chromosome 22, 121, Y are much smaller than chromosome 1. Thank you for correcting me. So there were many more germline mutations in males as opposed to females. Human genes were unevenly spread through the genome. So as I told you, in prokaryotes, it's very gene dense. But in the case of human genome, there were some regions which were gene rich, and there were some regions which were gene poor. So the regions which were mostly gene rich had a higher frequency of G and Cs. And you can probably tell me why. Can somebody think about a reason why would this be the case?
Can you repeat the
Why would
question, sir?
gene-rich regions be having more Gs and Cs?
Uh,
regularly transcribed. So for the stability, we required high GC content. So because GC have three hydrogen bonds, so that stabilize the DNA.
Okay. So could this also be due to the fact that if you have a gene, the mutation rate expected in a gene would be different from a region which would not be having genes,
Yes, sir.
right? So think about that part as well. All right. So, but what also happened during the process of this, from the process of sequencing, that they came up with different tools or bioinformatics tools. One of these tools was called the genome assembly tool, called FRAP, also called fragment assembly program. It's a computer program in which you can give raw sequences, raw DNA fragments, and it would piece them together like this and will give you a sequence contig or a continuous sequence that was composed of these individual fragments. All right. But during this process, it was very clearly understood and appreciated that we need to speed up sequencing. And here is an example of some calculations. So a typical Sanger sequencing machine, if many of you, I asked this question initially in the class, but none of you answered. So there was a video that I posted in the in the beginning of this lecture, which shows you how Sanger sequencing happens. So simultaneously in a Sanger sequencing machine, you can sequence approximately 96 fragments in about 12 hours. Okay. Now let us say that approximately the largest size of a fragment that you can sequence is about 1000 nucleotides, right? And that's the good quality of DNA that you can do. So that essentially would mean at any point of time, you would need, so if in order to sequence the entire human genome once, you would need to sequence 3 million fragments, right? If you are sequencing 3 million fragments, let us say you are running approximately 100 fragments in 12 hours. That means you will have to run the machine 30,000 times or for 15,000 days, right? Now imagine there are 100 such machines spread across the world. That would essentially mean that it will take you about 5 years to sequence the human genome, right? And this is only to sequence the human genome with one coverage. But we know that we sequence the human genome with much more coverage. So there were many more machines that were sequencing. And you can clearly see that that's why it took such a long time to sequence a human genome. Now this is not viable, right? I hope everybody can follow this calculation. Yes, no?
Yes,
Yes, sir.
sir.
Yeah. So therefore, there was a need to speed up sequencing. And they went back to the very basics of how sequencing happened. And that's why I was walking with you through how they actually perform shotgun sequencing. So they went back to the basics of first principle thinking, and they took the advice of mathematicians, chemists, and physicists. So these are the questions that they wanted to answer in order to speed up sequencing. The first question was, can we separate DNA molecules more efficiently? So earlier, the DNA molecules were being separated by the process of cloning. All right. Mathematics said that you can also do the same process by dilution. If you take your DNA molecules and you dilute them so much that you only have a single molecule, then that's also one efficient way of separating DNA. The next question was, can we increase the number of fragments to be sequenced? So now if you would take this process of dilution, then you would need to dilute a lot, right? So how would you actually capture such diluted parts efficiently? So they came up with the idea of micro compartmentalization. So you dilute samples in micro compartments so that each compartment is likely to have not more than one DNA fragment. All right. And then after this micro compartmentalization, you can do the experiments there. Second set of things started to happen with the use of chemistry. So if you use chemistry like fluorescence, you need to have a microscope which detects fluorescence in the molecule, right? Now, when you want to detect fluorescence in a molecule in the Sanger sequencing gel, you need to have certain number of molecules, right? So one question that people started to have was, can we actually reduce the number of molecules that we require in order to receive the fluorescence signal. So that is one. The second thing was, can we actually use something else as a proxy to detect nucleotides, to detect the identity of the nucleotides, which is not fluorescence based? And that is when chemists suggest that you can actually use pH, right? Physicists, on the other hand, suggested that you can actually use the physical properties of the enzymes that are elongating uh, the, that are elongating your polymerizing DNA. And this gave rise to the ability to look at the electrical change in electrical conductance across a protein when a DNA molecule passes through it, which is called Oxford nanopore, and also looking at how the vibration, how the different, uh, how fluorescence can actually be captured in a much more precise manner, which is what is being used in a machine called Pacific Biosciences. So I have highlighted three key technologies which I'm going to discuss. The first one is called Illumina, the second one is called Ion Torrent, and the third one is called Oxford Nanopore in the course of this course. If you're interested in other technologies, you can always come to me and I can share the relevant literature with you. So I'm going to end the class with quickly explaining how dilution works. Okay, so when you want to dilute, the problem that you're solving is a problem to efficiently distribute or select single molecules of DNA. So the principle is a highly dilute solution of DNA fragments can be partitioned or distributed into very small volumes of many compartments to obtain one fragment per compartment. This principle is called, if in mathematics, this is called the pigeonhole principle. Do you all know about the pigeonhole principle? Okay, if you don't
Yeah.
know, please read about it. It's like a homework for you. Now, once you have one fragment in each compartment, you can amplify with PCR or some technique all the fragment in that particular compartment only. And then you can sequence the fragments that are there in that single compartment using the appropriate method. So what does this do? This completely replaces, they have engineered a way to replace the need for cloning. Because what was cloning solving for you? Cloning was solving for you the problem that how would you take a fragment of DNA and select it and then amplify that specific fragment, right? So by dilution, you get rid of that problem. You are able to isolate fragments and then you are able to selectively amplify those fragments. So in order to do this, they came up with the idea of what's called emulsion PCR. So idea again, principle is dilute, partition, amplify, sequence. So PCR in an emulsion. So emulsion basically is a mix of two phases of liquids. So for example, if you take oil and water and if you shake it, then you will find these small emulsion bubbles, right? So that's the same idea. So what they did was they attached DNA fragments to a bead. Okay, now they encapsulated this bead in an emulsion, and then what they did was this bead, the DNA fragment that was attached, now this particular DNA fragment underwent PCR, only this fragment in the bead, in that particular emulsion. So now as a result of which, you are going to have these water droplets and these beads inside the water droplets, which are going to have copies of only one DNA fragment. All right. Now what you can do is you can take these beads and you can put them in picoliter wells, right? Each well is going to have a different bead, and now you can actually perform a sequencing reaction. So this is how it looks like, and you can perform sequencing reaction within these wells by doing some kind of fancy PCR or by doing some kind of sequencing by uh, by different kinds of methods. Okay, so I'm going to talk to you about how this thing works in the next class. So in the next class, I'm going to talk to you about the three sequencing technologies that I have introduced here. So we are already at slide number 50. So there are 27 slides more to go. Please be on time in the next class so that I can finish this particular part and we don't have to spill it over to the next week. All right, I'll stop here. Are there any questions?
Sir, the lab assignments